second speaker is Francesca Storici from Georgia Tech. And her lab has been kind of focused on fundamental mechanisms of DNA double-strand break repair. These are very relevant kind of to the genome engineering field. And she's investigated how cells use single-strand oligonucleotide donors. And in the last few years, her lab has published very interesting work on how RNA templates can also be used in the repair of DNA double-strand breaks.
Thank you. Thank you so much. And I would like to thank a lot also Fang and all organizers for inviting me for this exciting workshop. From experiment in budding yeast and later in also in human cells, we demonstrated that RNA sequences shown in red here, when embedded in DNA oligonucleotides or as RNA oligos, could serve as donors to repair double-strand break in a marker gene integrated in yeast chromosome. And the frequency with the RNA sequences was dropping, especially with increasing the size of the RNA. And this especially due to the high instability of this RNA molecule, because they were non-protected, so exposed to degradation. In fact, having a 3' tail of DNA without any homology to the break increase about 100-fold the frequency of the repair by this RNA oligo. So overall, this result suggested that the synthetic RNA sequences could serve as donors for repairing a double-strand break, and so there could be a direct transfer of genetic information from the RNA to chromosomal DNA. Moreover, with primer extension assays using templates containing different lengths of RNA tract, we show that to some extent DNA polymerases, in particular Pol delta of yeast, could use the RNA as a template, showing that in cells there could be some capacity to use RNA as donors in repair of breaks. Because most of genomic DNA is transcribed into RNA in cells, we wonder whether not just the synthetic RNA, but endogenous RNA could play a role in double-strand break repair in cells. And so if there is a way how we can have a flow of genetic information from RNA to DNA in a more general way than anticipated in the process of RNA-mediated DNA repair. And to address this question, we developed two experimental systems, the trans and the cis in budding yeast. So we have a marker here in the yeast chromosome, this history that's disrupted by the site for a nuclease where we can induce a double-strand break in this marker, and we want to see if an RNA shown in red can be the donor for repairing this break. And this is an endogenous RNA, so a transcript that's generated in the cells, and this comes from another copy of the history, and this also is inactive because it is disrupted by an artificial intron, which can be spliced only from the antisense, but not from the sense RNA. So our donor is the antisense, because this cannot be translated into the functional protein. But after splicing, this antisense, first of all, is different from the DNA that generated it. And so if this serves as a donor for the repair, this can restore the functional history, and we can have a phenotype, we can have His plus cells. Because yeast, like most of eukaryotes, has retrotransposon, they have reverse transcriptase, this can not only convert the RNA of the retrotransposon into a DNA copy, but potentially, to some extent, also any RNA could be reverse transcribed, as well as our history RNA. So then they can be repair driven by cDNA, so it will be, again, a DNA-DNA repair. So in order to block this formation of cDNA, there are some mutants that can be done, like this deletion of the suppressor of the TY, the spt3, which will minimize the RT activity in the cells. And so in this way, we would ensure that the only way how we can have a His plus is if we have a direct RNA-DNA recombination. So this is the trans system, because we have an RNA generated in one locus that is the donor for repair break in a homologous but different locus. In the cis system, we move the site for the nuclease in the middle of the artificial intron. So we have just one allele of our marker. And here we look if the RNA can repair a break in its own DNA, so in cis. And so this this construction was mainly done by Inshan when she was a graduate student in the lab. And so after we induce the expression of the antisense and the double-strand break, we are looking if in these two systems can we get His plus cells. And so with work done initially by an undergraduate, Katie Ashley, and then lots of work by Hava Keskin, a PhD student in the lab who just graduated. So we found lots of colony His pluses for the trans, especially also for the cis, more for the trans, and likely because in the trans system, the RNA can be continuously generated instead of the cis. As soon as there is a breakage, there cannot be more RNA produced. But everything is dependent on the sp3 because we have no colonies that are generated in this mutant, so suggesting that this is all a cDNA-driven repair. So if we wanted to see an RNA-directed repair, there would be a formation of an RNA-DNA hybrid, which would be the target of ribonucleases H, type 1 and type 2. So we knock out the type 1 and the type 2 as a single or both together. And we saw that only when we eliminated both RNase H's, we could see colonies even in the absence of the sp3, only in the double mutant. And these were more abundant for the cis than for the trans. And so this is what we think is the direct RNA-mediated DNA repair. And why this will be more abundant for the cis than for the trans? Likely because in the cis system, the RNA is nearby to the breaker. So in cis, the vicinity of the RNA would facilitate the repair by RNA. And so this mechanism of a more efficient RNA-templated repair in cis than in trans, due to the vicinity of the RNA to the target, is supported by our experiment of aptamer-driven gene targeting, which we have a donor single-strand DNA molecule. And if this is brought to the target site, in the vicinity of the target site by an aptamer sequence that has been selected to bind to the nuclease that makes a double-strand breaker, this stimulates the gene targeting up to 32-fold in yeast and 16-fold in human cells. So our His plus colonies that we obtain in the absence of both RNase H's and also when we minimize the reverse transcription in our cells of the cis system, they have perfect history sequence when we sequence them. And also the history is present in the expected genomic locus. When we extract the genomic DNA from the cells, we digest it with restriction enzyme, and we hybridize with a probe specific to the history. So we see the expected band, so suggesting that we don't have, I don't know, integration of some cDNA or transposition elsewhere in the genome. So from a larger band, we have a little smaller band because we lose the site for the intron and the nuclease when we have precise repair. So suggesting that this would be a repair maybe due to homologous recombination, or potentially could be an end-joining mechanism, but we excluded canonical non-homologous end-joining as well as microhomology mediated end-joining because our His plus colonies in different backgrounds, including in the absence of the RNase H and the sp3, where this should be the direct RNA repair, they all require the splicing to occur because when we make a little deletion in the intron to eliminate the 5' spliced site, so there cannot be splicing, we lose all our His plus, so suggesting that so it cannot be an end-joining because that would be independent from whatever happened at the RNA level. So again, this supports the fact that this colony that we observe here will be driven, so requires a donor, requires the RNA, so likely in an homology-driven mechanism. And in fact, we do see dependence on the recombination protein RAD52, which is important for annealing of single strands and also has a function in loading the RAD51, the RK homolog, to process the break ends. But we see reduction in the frequency of RNA repair when we knock out RAD52, but not when we knock out RAD51 or RAD59 has some homology with RAD52. And so when we don't have RAD52, frequency goes down for RNA. If we provide more RAD52 in this experiment, instead the frequency goes up. And even when we use the truncated RAD52 from Saccharomyces cerevisiae or human, which has only the N-terminal, which is the part more important for the annealing. And so the C-terminal is one responsible for RPA binding or for binding to RAD51. And so these overexpressions show increase in the frequency of RNA repair, both in the presence or in the absence of endogenous RAD52 in the cell. So this data suggests the RAD52 is a key player in an RNA-templated repair. And proteins that have a role in processing the ends, in resection, so in degradation of the 5' ends of the breakers, such as the site 2 and the exo 1, when we knock them out individually, actually we observe some increase in the frequency of the RNA repair, so suggesting that at least efficient resection may not be a strict requirement for RNA-mediated repair. So with this experiment that we just got accepted in molecular cell in parallel with our experiments of biochemistry conducted by our collaborator, Alex Mazin at Drexel University, we show a distinct new function of RAD52, and both for the human in yeast RAD52 purified protein, we found the capacity of in performing inverse strand exchange. So the classical forward strand exchange reaction, which is like the strand invasion mechanism, works with a recombinase that will be here in green. It could be the RAD52 or RAD51, so that is loaded on a single strand DNA. And this catalyzes the exchange with the double-strand molecule. So this is the forward strand exchange. And the reverse is the opposite. So we have the recombinase in green. Now it's on a double strand. It's a broken double strand, not a circle. It's a broken linear double strand. And with a little tail here at the 3'. And this catalyzes the exchange with a single strand. So this is the inverse reaction. And so RAD52 works much better in the inverse than in the forward reaction, and not only when the single strand is DNA, but also when it is RNA, both the human and the yeast. Actually, the human works even better than the yeast. And moreover, we found that this reaction works with this 3' tail, but even if we reduce the length of this tail, or we eliminate this tail to have a blunt, to mimic a blunt end, like here, so we can see here different size of the tail, this reaction, this inverse strand exchange reaction still occurs or can occur even with a blunt end. So this sort of parallel the in vivo results, maybe suggesting that RAD52 could mediate an inverse strand exchange even in condition of limited resection. And just a few experiments to show some involvement of what could be the polymerase that use the RNA as a donor in the process of repairing the breakers. So we saw that there is not a requirement of a bona fide reverse transcriptase because we already show that RNA repair can work with the mutant in the absence with the sp53 mutant when we minimize the RT. But here we have a strain from Paradoxus that doesn't have retrotransposon. And we see that in this strain, we don't have any cDNA repair compared to the strain that has the retrotransposon, but we're only left with the RNA repair when both RNase H are mutated, suggesting that RT is not required, but when we provide more RT to the cells because we overexpress it from the yeast or from murine leukemia virus, we have an increase instead in the frequency of repair. So these are our model for repair, how an RNA transcript can mediate repair of a double-strand break. So in an actively transcribed DNA that experience a break, we can have RNA as a donor, depending if we're in condition of limited resection or if there is more resection. So we have limited resection, then the RAD52 recombinase can catalyze an inverse strand exchange reaction. If there is more resection, instead there will be a strand annealing reaction catalyzed by RAD52 that we show before. The RAD52 has this capacity. But as soon as we have this RNA-DNA hybrid, at least in our system, this will be attacked by RNase H, so we have to eliminate those in order to see this repair occurring. And so the repair then would proceed with without an RT in the first case, because there would be just a maybe very short gap to be filled, so very little requirement of an RT function. Instead, if there is more synthesis, then likely an RT function can facilitate this repair to occur. So then in this way, we can restore the break and have repair the break. So I would like to thank you very much. And here are the people of the lab. And also thank the collaborator, Alex Mazin and David Garfinkel, for many plasmid, and our sponsor for this work, mainly NIGMS, NSF, and HHMI. Thank you.
Francesca?
So in situations of limited resection, have you tried to increase that the probability of that happening when you delete Q70, Q80?
So we are in the process of studying how this mechanism can be regulated in different stages of the cell cycle, because, for example, in the G1 phase, it's supposed to be less resection. And so to see how, what could be the efficiencies in different stages of the cell cycle. In the assay that we utilize, this requires formation of colonies, so cells are actively growing, so we are making a different system. Also exploiting the CRISPR-Cas approach, which will make much more versatile the way to study double-strand break repair by RNA.
And do you think RPA is involved in this, or single-strand binding protein?
So RPA actually does experiments in vitro. We saw that in the presence of RPA, the inverse strand exchange reaction is actually stimulated with RNA, not with DNA, but with RNA. It actually brings it up. And in vivo, we haven't done assay with the RPA, because RFA probably are all essential. I mean, we can make some mutants, but we haven't done that.
And one last question. So in terms of transcription, do you have a sense of what kind of genes would likely use these mechanisms in the cell, like highly transcribed genes or lowly transcribed genes?
Right. It definitely requires transcription, because the transcript will be the donor. So that's definitely actively transcribed DNA. So an idea that we had was that this could be a system the cell could use in condition where there is no sister chromatin, so maybe non-dividing cells, so they don't have any other donor for repairing a break, because if there's no sister, it could be the homolog that may be far away, more complicated system. So if there is no cells are not dividing, then if they are in G1 or G0, the only way to repair a break would be with non-homologous enjoining or microhomology enjoining, so that would be very mutagenic. And so if there is an RNA there in the vicinity, that could be, why not using that as a donor in a homologous recombination mechanism? So potentially that could be where cells could use this.
Thanks.
One final question.
All right, really quickly, have you looked have you looked to see if this the repair off of RNA, the mechanism, requires any contribution or has any genetic dependencies on the transcription associated proteins or RNA splicing associated proteins, or things not traditionally associated with double-strand break repair?
Right, right. We are, yeah, with the process we're doing that, I mean, we show the splicing requirement, but for us, the splicing is just a tool to distinguish the RNA from the DNA. So that, and yeah, we are in the process, but we don't have any candidates now. Okay, thank you.